So, good afternoon and welcome to our seminar, Challenges in Forest Ecology. So great to see so many people already online. So our speaker today is Lars Vesterdal. He's from Copenhagen University. He's Professor of Ecosystem Processes and Soil Carbon. A quick sketch of Lars. He did a PhD in the late 90s at the Royal Veterinary and Agricultural University in Denmark. He then moved to the Danish Forest and Landscape Research Institute, where I guess he stayed about 10 years or something along that nature. And since 2014, he's been at the University of Copenhagen, where, as I said, he's now full professor. Lars's interests are pretty much soil carbon and nitrogen stocks in soils, also greenhouse gases, and probably more recently, factors affecting soil carbon stocks. So Lars has, again, a pretty remarkable H-index of 68. His work has been cited over 20,000 times, including 2 papers, which have been cited over 1,000 times, and one nearly 2,000 times, which is definitely a bestseller by any stretch of the imagination. And he also has the longest title we've ever had. So Lars is going to talk today about soil and ecosystem carbon sequestration over 50 years in resampled oak and Norway spruce afforestation chronosequences on former cropland. So great to see you, Lars. It's your video, as it were, your screen, your stage, and off we go.
Thank you very much, Doug, for this very flattering introduction. I'm really happy to be here today and really enjoy being in this distinguished company here. Thanks for reading up the long title, so I don't need to do it myself. But it's a pleasure to be here to tell you about a really long-term study that, well, goes beyond almost my entire career here on afforestation in former croplands. And this work is done with a lot of cooperators, and particularly I want to also highlight my recent PhD student, Ming Yu here, who has really taken this off in the last 3 years. But why do we need to know about afforestation? See if it can change this. Yep. It's long been highlighted that afforestation can contribute to as a major climate change mitigation measure. There was this paper a few years ago about the global tree restoration potential. Here at the European level, we have the 3 billion trees pledge under the European Green Deal, and not least in minor Denmark, we have a very huge initiative now to plant 1 billion trees in the next 20 years and increase our forested area by 250,000 hectares. That is equivalent to converting 10% of our farmland into forests. So at different levels, we need a lot more information about, well, what do we actually achieve by this climate mitigation measure, and particularly from my point of view, of course, what do the soils contribute in this context? And what do we really expect after cropland afforestation? We are currently working in this Silver Nova project on how to best transition croplands into something that is at least or will as fast as possible be equivalent to what we know as ancient forest ecosystems, these forests that have a long continuity. But we are often seeing that a lot of these forests planted on former cropland, they are different in forest structure and microclimate, biodiversity, soil processes, and soil properties. And today I really want to highlight the soil processes and particularly the carbon dynamics and carbon change in the soils. What can we expect in these systems? How fast can we expect that we reach levels that are more comparable with ancient forest soils? And which processes will be in play here? So just a quick look to what these soils look like. Here we have an example of a cropland and a forest soil profile dug within 100 meters here in Denmark. And the main question is, of course, how fast will the carbon stocks, but also the stability or the persistence of this carbon change over time, because we obviously want the carbon to stay in the soils for as long as possible and stay out of the atmosphere. And well, visibly looking at this, of course, there is this transition from this very intensive management with the plow layer that we see here in the left photo to this much more developed or heterogeneous soil profile to the right, where we also note the development of the forest floors happening over time. So where can we study this? Here is it that we have been able to address this question in time series of afforestation chronosequences in a forest area or a developed afforestation project just outside Copenhagen, 15 kilometers west of Copenhagen here in Denmark. And what I'm showing here is an aerial photo of what this area looked like in 1954. The only tiny forest area is visible here to the, see if I can get the marker to work. No. Here, the only forest area, let's just go back. The only forest area here was this very small patch of 5 hectares. Every part of the landscape was used at the time for intensive agriculture because these are among the very best soils for agriculture in Denmark. Here you see a lot of different dots also, and when I now change the time to what it looks like today, you see that a lot of this area has been now afforested. We still have areas with permanent grass also in between here, but the entire area here is now has changed from cropland to either forest or permanent grassland. And what you see here are permanent plots of planted Norway spruce, and oak, and then as reference we have the 200-year-old forest here to the left. We have a permanent grassland also planted on former or established on former cropland, and then we have cropland references still with ongoing typical cropland agriculture here in Denmark. We have been able to follow these areas first time from 1998, then a new inventory in 2011, and another one recently in 2022. And in this area, we have these time series chronosequences. We have one in oak, which originally spanned from oak that was 5 years old to 30, and we had Norway spruce from 1 to 30 years of age back in 1998. And then we had the old forest as reference here. What does it look like, the soil here? It's important also to understand the context that this study has been done in, because it's really not what you would call marginal soils that are often set aside for afforestation. Here, the main driver was actually to create a recreational area for Copenhagen. It's very nice cropland soil. It's fertile loam with quite a lot of clay for Danish conditions, and it's developed from calcareous till. So it's very fertile and very rich conditions, also in parent material, not only because of the cropland use where there's been fertilizers and liming involved over time. And This just to highlight the long-term nature of this study. I put in a photo here to demonstrate that this is really long time ago when we started working here. First study was back in 1998, as I said, with the first kind of report of the chronosequence. But then we managed to do a resampling back in 2014 with Teresa Gomez here as the main driver, and Teresa did a lot of work on to follow the changes in the chronosequences. And now we have the opportunity to had an opportunity to do a third resampling under the guidance of Ming-Yu here in the bottom here. And here I'm showing what the design is really about. We have from one, you would say, one matrix of chronosequence here with stands of different ages over time, initially 1 to 29 years. And now in 2022, these stands have moved up. When we come back to resample them here, they are now in the age class of 25 to 53 years since afforestation. So we have a combination, an integrated assessment along chronosequences, but also with resampling within the specific stands 2 times here. We still have resampling of the cropland conditions, and we have resampling of the old forest conditions as well along the way here. Here I want to focus firstly on the soil carbon stocks. That's the main topic of my talk today, but I also want to scale up to the ecosystem carbon stocks, including the biomass carbon sequestration in the study. And next, in the end, I want to zoom in and also address, well, where is this carbon actually sequestered in the soil? In which parts of the carbon? Is it in the what we use to term mineral-associated organic carbon, or is it particulate organic carbon? And finally, I want to zoom in on what is the origin of this carbon that goes into the different carbon fractions by looking at the contribution of microbial necromass to the new carbon we see in the soils. So what did we do? Here we are. So in all 3 periods here, we did similar sampling of the forest floors here to the right with the usual frame, and then we did coring to the as shown to the left, and separated the top mineral soil equivalent to the plow layer into 3 different segments, 5, top 5, 5 to 15, and 15 to 25 centimeters. We quantified then the carbon stocks later on based on, of course, carbon concentrations and bulk densities, and also corrected by the equivalent soil mass method, because the bulk density change over time in all afforestation projects when you stop managing the soil, and the soil tend to loosen over time. There's not a lot of time to go into these details. We can always discuss that in another opportunity. We also did woody biomass measurements to be able to model the total tree biomass carbon stock above and below ground in these plots. The recent thing, or the new added components, are then the size fractionation of the organic carbon in the soil into fractions that we can at least interpret as particulate organic carbon and mineral-associated organic carbon, and then the work to also address the contribution of microbial biomass to the soil carbon stocks here. So what did we find here? Now I'm going to show a range of graphs here where the legend is the same in most of them. And what we see here now is the integrated effects or the integrated measurements of the different chronosequence, but also of resampling. So you see symbols in the graphs here from the different inventory years. And in this case, we also see here different curves of carbon stocks in the forest floor for spruce, Norway spruce, and for oak here. To the right, we see the old forest reference, the 200-year-old forest, resampled with the same along the same inventory years. And what we see is that we generally see forest floors stabilizing. They don't continue to grow, at least not in all plots. For oak particularly, it's very limited. What we see here, after about 3 to 4 tons, the forest floor carbon stabilizes. In spruce, it's a bit more scattered, but most of them also tend to stabilize, with the exception of mainly one stand up here. So forest floors tend to stabilize over time, and you can also see that the old forest indicates that we cannot expect a lot more to happen in, at least in broadleaf tree species here. Turning to the mineral soil, I'm showing here the aggregated effects or the aggregated carbon stock change in the mineral soil to 25 centimeters. And here we see more similar trends in the two tree species, but we do see there are slightly higher rates of change in spruce with 0.4 tons per hectare per year compared to oak. And shown to the right, we also note that, well, there's still a gap to reach the levels in the old forest reference here. To the left, you see now the cropland reference here included with about 40 tons of carbon per hectare in the same layer here. Notable is maybe that the grassland, the light green colored symbols here, is doing pretty well in this context also. We only have one major field here, but it tends to also show increases in soil carbon over time here. Summing up for the soil here, the summed effects on carbon stocks is like this, that we see a kind of widening of the difference between the species because the forest floor is adding more to the spruce soils here. And notable is that spruce is actually approaching the level of the old broadleaf forest here after about 50 years, whereas oak is still lagging behind in terms of carbon stocks. Now I want to try to show some results based on the repeated sampling, because here we have the opportunity to also look at the carbon sequestration rate for 2 periods, because we have repeated sampling. We can look at what happened from 1998 to 2011 and 2011 to 2022. So here I'm showing now the rate of change on the y-axis. And for the forest floor here to the left, you can see that we start out with some appreciable rates, about 0.5 tons per hectare in the spruce when stands are really young and developing. But after 30, 40 years, they decrease and they reach levels that are around 0. So indicating, again, confirming that we don't see a lot of change after 30, 40 years in forest floors. The forest floors are contributing initially, but then the sequestration effect levels off. To the right, you see mineral soil, what's going on there. And here it's basically mainly in the spruce we see an increasing rates over time here. So the spruce soil is still, you could say, increasing in rates and average. A lot of scatter because these resamplings are also with a lot of uncertainty. But notable, you can see that oak is not doing a lot here. It's just around 0, and there's no significant development in rates for the oak stands so far. So spruce is again, you could say, indicated here as the most efficient, you would say, carbon sequestrator in the soil. Summing up with the different compartments in the ecosystem, I'm now here also adding biomass carbon to the picture. Here we see the total ecosystem, the green curve here. We see the biomass carbon alone, the purple curve, and then we have the soil in the bottom here. And to the right, you see the same data for the old reference forest. And again, showing up here that soils are increasing slightly more in spruce, but the biomass is really, the biomass carbon sequestration is a lot higher in spruce. It's well known, spruce grows faster than oak, so it's not rocket science. But it shows that it's really the factor that also makes the spruce ecosystem here in being the most, you could say, efficient post-agricultural system compared to oak. All this carbon is not new, because going back to this one, of course, we have a significant carbon stock in the soil. So the final synthesis here with the carbon stocks is to address what is actually the new carbon sequestered in these ecosystems after 50 years. And here I'm again showing that, well, the most efficient system is in Norway spruce, 147 tons of carbon per hectare after 50 years versus 82 tons of carbon per hectare in oak. But the most interesting thing here is actually that we see when we look at the distribution of the carbon in the ecosystem, it's surprisingly similar. The distribution of the new sequestered carbon, with about 85% of the carbon sequestered in biomass, 5% in the forest floor, and about 10% in the mineral soil until 50 years here. So this could indicate at least that the biomass and the inputs from the trees could be a main driver also for the development in the soil, rather than very different decomposition rates or different, say, stabilization in these systems here. We also know from before that we do not see huge differences in the soil respiration, in the carbon reactivity in these systems when we look at the mineral soil. So it seems really here that the biomass and the biomass development here is driving also what is going on below ground to a large extent. Now we wanted to try to, to dive a little bit deeper into what's going on with the soil here. So now I'm going to talk a little bit about the soil organic carbon composition, and finally the microbial and necromass assessments here. First, the carbon fractions. We did a simplified carbon fractionation procedure here, because it's really a lot of soils, it's a lot of stands, and therefore we chose the quite, you could say, often used size fractionation here over the size density fractionation, which is a lot more laborious. And also to limit the number of samples, we focused here on the 1998 samples and the most recent 2022 soil samples we have from the chronosequences. But the procedure here was to get 3 size fractions, a size fraction less than 53 micrometers, which is mainly silt and clay, and can be interpreted as a proxy for being dominated by mineral-associated organic matter. And then we have 2 fractions, which are the larger, or the sand fraction and the silt microaggregate fraction, where we have the larger POM and the smaller or more decomposed POM in this fraction. But altogether, these 2 fractions would represent the particulate organic carbon. So what did we find here? Here I'm now showing the development in first just in the carbon concentration in these different fractions. You see in the top some microscope photos of the three fractions here with the smallest fractions less than 53 being here in our interpretation at least a proxy for mineral associated organic carbon. And similar to what we saw before, we see that, well, we get more carbon, and we get more carbon in all three fractions, as it's shown here. If we first look at the mineral associated organic carbon, or the smallest size fraction here, we can see that there's still a little bit of way to go to the old forest reference, but we see an increase from the cropland situation here, also in carbon concentration in this fraction. In the two coarser fractions that are more representative of particulate organic matter, we see that the spruce is different from the oak, and we see a lot faster increase or greater increase in carbon concentrations in spruce versus oak in this case. Oak is kind of getting to the level of the broad-leaved old forest here in the country. But again, we can see that the particular fractions here, or the carbon, increases some very minor concentrations in the cropland situation. This is showing again, okay, we get more carbon into the soil and into all three soil fractions, but what is the share of the carbon or the change in the share of carbon in these fractions? And here it gets maybe more interesting, because we can see that we have a large decrease to the left here from in the smallest size fraction. In the cropland situation, we have a very high percentage in this fraction, about 80%, and it goes all the way down to, well, close to 20% in spruce, maybe 40%, 40% in the oak. And in the old forest reference, we also have about 40% here. On the contrary, we see that the share increases with the particulate organic matter, or the coarser fractions here to the right. And in the coarsest fraction, again, the spruce separates from oak. We get more particulate organic carbon, as it seems, in the spruce. And it also goes beyond what we have in the old forest reference, where we are at a very similar level to the oldest afforested plots here. So there is quite a transition going on in the fractions of carbon. The last results I want to share here is now where the results on, or at least an indication of, where does this carbon come from in the soil? Is it plant? Is it a plant origin, or is it of microbial origin? And here we focused again on just the samples from 1998 and 2022 to have the longest time span in repeated sampling. And we have, as before, I forgot to mention it, but this was for the top 5 centimeters of the mineral soil. And we also limited it to the two most deviating size fractions here. So, and we used amino sugar biomarkers here, glucosamine and uramic acid, and that enables us to, well, look at what is the kind of aggregate microbial necromass carbon, but also to separate the fungal and the bacterial necromass carbon based on these biomarkers, these amino sugars. And yeah, there's a lot of other things. It's a lot of work and a lot of proximate analysis. So yeah, it's not trivial analysis to do in terms of labor. And here is what we found with respect to the microbial necromass. And here I'm showing the proportion of microbial originated carbon in the fine fraction here and in the coarsest fraction here. And there was no significant change along the chronosequence here. There is a slight increase. It was not quite significant, but actually we do see a decrease when we look at the cropland here. We did have more contribution of microbial necromass, and we seem to have less contribution from microbial necromass in the old forest. And that also goes for the course of action here. So this indicates that we would expect over longer time spans that we have more plant, more carbon or plant origin contributing to the carbon in these systems versus microbial. Looking at in more detail, however, we did see that there seems to be more happening when we look at fungal necromass here and bacterial necromass here in the bottom. The two top graphs here show the fungal necromass in the fine fraction and in the coarse fraction. And here we see that there was an increase indicated for fungal biomass or fungal necromass carbon in the fine fraction over time here. It's not a lot, and we also see that it's not appreciably higher when we look at the old forest, but there is some slight increase here. When we look at the particulate organic matter here, or at least the coarse size fraction here, we see only in oak that there was signs of some increase. But the curious thing is a little bit that we have this jump from cropland to afforested plots here already happening very fast. And then this slight increase again in fungal necromass contribution here. So there's still some work to do here, maybe to explain these things. In spruce, we didn't see any change at all happening here. And looking at the bacterial biomass or bacterial necromass contribution, there was absolutely no change over time, except that we did see more bacterial necromass contribution in the cropland here in general. We also saw here that oak seems to have a greater contribution of bacterial necromass carbon compared to spruce, but no clear temporal development over time in this. So we're getting to wrap up here. The main findings here was that, well, with respect to carbon stocks and sequestration, we did see that soil carbon increases along with biomass carbon over these 50 years, more in spruce, in Norway spruce than in oak, as representatives here of conifers versus broadleaves. Interesting was that the soil carbon sink, at least in this context with these fertile loamy soils, then the soil carbon sink shifts from the forest floor to the mineral soil after 3 to 4 decades. The ecosystem carbon sequestration was higher in spruce, but the relative distribution of carbon in the ecosystem compartments was surprisingly similar between the two tree species here, as shown to the right here. Looking at the carbon fractions in the soil and the sources for this carbon, we saw that, well, the general increase in bulk soil carbon is reflected in both carbon fractions in particulate and mineral-associated carbon, but there is a greater proportion of the new carbon in the particulate organic carbon. In spruce, we are almost up to 80% of the carbon which is in the particulate organic form in the top 5 centimeters here. We see a higher proportion of plant-derived carbon in forest and cropland. The age trend was not really significant there, but we do see an increasing contribution of fungal necromass to mineral-associated carbon, and at least for oak, there was a tendency that also the particulate organic carbon could be contributed by fungal necromass to a greater extent over time. When these systems develop from really being cropland, having a strong cropland legacy, to being more close to older forests. So I just want to acknowledge the really hard work done by Ming and also by Teresa before here, and all the other Vesterdal collaborators I've had during the last 20 years here. And then thanks a lot to all of you for listening, and well, I'm hoping for some questions.
Great, Lars. Thank you very much indeed. Very, very interesting. So we have now questions. What we normally do is we write the questions in the chat box. If you have a desperate wish to ask the question yourself, of course you're more than welcome. Lars, you can see the chat box, can't you?
Yeah.
Yeah. Okay. So the first question is from Cindy, which was apart from what were forest floors of both species more or moder? Were both species ectomycorrhizal? And do you think most of the fungal necromass was mycorrhizal fungi? That's 3 questions actually, but
yeah,
questions.
A lot of questions. Great. I'm just opening the. Yeah, well, first, yeah, both 3 species here are ectomycorrhizal. So, and oak is different from spruce. That's evident from here also. But when we look at a range of other broadleaves, well, it's maybe in the area closer to spruce in some respects. They're both ectomycorrhizal, and yeah, I would really expect that that quite a lot of this fungal necromass could be yeah, mycorrhizal hyphae driven. We have had a student project going on this, but it didn't show very strong developments over time, and we need to do more on this. We are doing finishing also eDNA analysis in these chronosequences, and I hope we will get a little bit closer to also to the functional traits of the fungi here, because would be really cool to get closer to this. And then there was a third question as well. Let me just see if I can find it here.
I think that was.
Yeah, I think that was it. Yes.
Yeah. So the next question is.
Oh yeah, maybe that was one. Yeah, more on motor. Yeah, maybe that was the one I forgot. They are the oak is basically not accumulating anything. What you see here on this picture here, is sort of representative. You basically have leaves and a little bit of F material underneath, fragmented leaves and so on. So it's very limited, whereas the spruce do develop maybe something which is a little bit more motor-like, but again, very loose material, and in some cases just loose needles on top of mineral soil. So it's very different from some of the, you could say, humus forms that we see if we are on sandy soils in this case.
Great. Okay, the next question, I think, is: did you notice changes in pH during different samplings through the years?
Yeah, we did. It's the pH is one of the most responsive variables here. So that is also supporting that we will see a change in from, let's say, bacterial dominated to fungal. In the croplands, it's about almost 6, the pH maybe 5.5 to 6, and then we go down to about 4.1 in the top of the oak and spruce. Surprisingly, the oak and spruce did not and has never differed significantly in pH over time. We had always, I mean, it's always textbook knowledge that conifers are acidifying more than broadleaves, but it just hasn't showed up here. Maybe a slight tendency for oak to become lower, or for spruce to become lower, but still not significant. But that is supporting that we will see a change we see this change in microbial communities, definitely.
Very great. The next one must be from Martin Valtera, which is, isn't the higher spruce contribution in soil organic carbon sequestration due to the different root distribution between the tree species?
That's a really good question, and I would really like to know more about it for sure, because it's definitely true. We have a higher root biomass in the top there in spruce than in oak. We know this from tree species experiments. We haven't really analyzed the roots specifically here, which is still you could say something which is waiting to be done in greater detail. But it's definitely true we could have more root inputs in the spruce than in the oak. However, again, we don't see apart from we don't see, you would say, a proportionally higher storage of carbon in the soils under spruce when we look at the total ecosystem distribution. That's still the curious thing, I would say.
Okay, great. And then a slightly different question. Can I ask you where you plan to share these findings outside of the academic community? For example, do you have any forestry partners or landowners that are involved and interested in the findings from these studies?
Yeah, we do have this, and also not well. It has been quite variable over the years. I mean, since 1990, we've actually had an ambition in Denmark to increase or to double the forest area. What is happening now? What I talked about is that this is really put on fire, you could say, because they have created this hugely ambitious goal just until 2045. And this makes the policymakers and people also interested in this, and also, you know, from everything from, you could say, policymakers and interested in ecosystem services and to national carbon reporting because of this major land use change that we are seeing here. But they don't participate specifically in the projects we have, I would say, but they are generally quite interested. And when we have seminars, there's quite a good audience also from the policy side.
Okay, great. And then from Andreas Schindelbacher, there's two questions. How is Norway spruce doing in Denmark? Here in Austria, I would not really recommend afforestation with spruce.
No.
That's
Neither would I. So I totally agree. This is, you could say, for historic reasons that, well, when this was established starting back in end of the 60s, well, spruce was just the main choice in Denmark. And even this site is among the least suitable for spruce, even these kind of quite heavy fertile soils, where spruce is even less stable than in other, you could say, climate change challenged sites we have in Europe. So it's more because we have this opportunity, and if we could create a decent chronosequence in spruce here, that we have used it as a representative of conifers. In some of the sub-studies we have recently also out here, we have a smaller chronosequence added also in beech in the same site recently, but with less intensive soil sampling, and only done here since 2022. So we are trying to get information on more, you could say, climate change adapted species at least.
And then the second part of the question was, did you also sample some cores deeper than 25 centimeters?
Mm-hmm. Yes, we did.
Oak roots may go deeper. Yeah.
Totally correct. And we have done samples we have sampled down to 50 centimeters, and basically didn't see anything happening down there. But we have used some of these deeper samples also for equivalent soil mass correction. For the time being, but we have them in storage for you could say later comparison with changes later on. But so far we see the main change, and I didn't show this right now, but yeah, I can just quickly maybe show this graph here where things are going on. I have this figure here also that you see now, where you see the three different salt depths here: 0 to 5, 5 to 15, and 15 to 25. It's just carbon concentrations here, but you can evidently see that most of it is going on with carbon in the top 5. Then we do see something starting in the maybe in the 5 to 15, and then there's absolutely nothing going on yet in the 15 to 25. So it's also quite a lot about redistribution on top of increasing the carbon stock.
Okay. The next question was from John Healy, is what factors will have the biggest influence on how well these results extrapolate to other soil types?
Oh, yeah. It's a very good question, and it's very different when we look at sandy soils here in Denmark, I could say, because we have done studies also some time ago in a sandy chronosequence, smaller chronosequence, only spruce though, but to compare with this one, and it's a tremendous difference in, you could say, forest floor accumulation, but not very clear signs that a lot of things happened in the mineral soil again. But I am very humble in terms of this, and I'm not saying this is in any way representative of Denmark. And this is where we particularly need, for, you could say, to satisfy our policy needs, to get better information from other, you could say, textual classes, or at least other conditions here.
Yeah, okay. And then Cindy Prescott again with a very specific question. Grasses seem to be better than trees at generating mineral associated organic matter. Do you have comparisons with grasslands or pasture in addition to cropland and forest?
Yeah, I don't have any more chronosequences in the grass, or so this is the best example I have actually in terms of setting it aside. And I know in Denmark they have I know people, my colleagues in agriculture here, who are more into grasslands. It's only recently, because in Denmark we have a tradition of tilling everything we can till. So this thing about permanent grasslands is only recently attracting interest, also because grass has been a kind of a crop, an interim crop in the normal rotation or in agriculture. So they are struggling actually to find good data for the effect in agriculture when we convert to permanent pastures or to permanent mown grasslands. But I totally agree, and I think it's in line with most literature that we see that grasslands are quite efficient in sequestering mineral-associated carbon, and in general also quite efficient in sequestering bulk carbon also. Yeah.
Yeah, okay. There's another one, which was, did you also study nutrients in the ecosystems? If yes, would it be interesting to see and share your publications? And I am working on ecosystem nutrients of different Mediterranean oak systems. So
yeah, that would be nice. And so please just send me an email, because we have an older study, but now we have a brand new one coming up also on the nutrient development in nutrients, also in carbon in carbon heterotrophic respiration, carbon reactivity, also on pH and phosphorus, what's happening there. So just send me an email about this.
okay, that's not a question, but there's also a statement that we're working on nutrients in different pine systems in the Mediterranean. Obviously, the Mediterranean is the place to be.
Yeah.
Looking at a very dark night in Brno, I can tell you it could only be better. So,
and snowstorm here in Denmark.
So, but I just have one. We still got just a little bit of time. I'd like to ask a question myself, which was, did you did you try and quantify losses of old soil carbon relative to the new soil carbon? How it is, because they start with considerable carbon stock. So what happens?
I would love to do that, but unfortunately, I mean, we don't have this maize cropping as they have in many other countries where you can do isotopic fractionation of the old carbon versus the new carbon. So it's complicated here. I would really love to hear if anybody has suggestions about this, but because it's really elegant what you can do when if you had continuous maize cropping, as in Southern Europe, where they have also where they can demonstrate the new versus the old carbon, because that would be the really great test of this of the new versus the old. What's happening also? How is it decomposing the old carbon, and what is the amount of new carbon coming in?
Yeah, okay, I'm sure that's a really important factor, actually. The other thing was that you get this when you first plant spruce, you get in your forest floor fraction big variations in the amount of carbon stored in spruce compared to in oaks. Do you know why that is? Why does it suddenly vary so much?
That's also really a difficult one, because we have really pondered about, and I'm going back to it here. This one, yeah, where we have it up here, and it's really it's a bit of a mystery to me, because it's within very it's within hundreds of meters distance. Here we have some of these stands, and this one, which is you could say one of the most outlying one here, is it's the oldest one also. And what we cannot totally rule out is, of course, that we have some kind of slightly different afforestation management in terms of how it was fertilized, how it was limed, or things going on there. But we don't see a huge difference in you could say, soil chemistry, in pH and so on. So it is a bit of a mystery in general why it's continuing, because the trend generally here is that actually some of them are actually decreasing in carbon in the last inventory, and we have very thin layers of just with needles. This is the only stand where we have a decent kind of development of a forest floor, and some as a very, you could say, crude hypothesis is that we've had some really dry summers in Denmark, and in some of these spruce stands we have very high leaf area index, and it actually since 2018 it stayed dry for, even during winter period. It was never re-wetted, because when it first dries out, it's just water repellent, this organic matter in these forest floors. So we have observed that it's been, dry in February in large patches also in this stand, and maybe that can partly explain maybe why some of there's been really this increase going on here. But then we should see a decrease next time we come there, and now things have stabilized more with the moisture also. So yeah. It's a good question, and shows also the limitations of chronosequences, what's going on. But what we can see is that it's not only because it has also, when we look at this plot, the stand specific rate has just been higher here in the recent years, but difficult to explain.
Great. Well, I can't see any other questions, Lars, so thank you very much. A very interesting talk. I'm sure everybody enjoyed that. Great contribution to our series. And again, thank you very much indeed. For those of you who know, our next speaker on the 1st of April will be Richard Norby, and he's going to talk about the Amazon face and the hypothesis generated from previous faces. So he'll put up something about all different face sites. So once again, Lars, thank you very much. Great to see you.
Thanks a lot for my side. It was a great pleasure, and great to see a lot of familiar names here. So thanks a lot for this opportunity.
You're welcome. Bye-bye.
Bye-bye.